students, myself Shweta Gandhi, Assistant Professor, LG Institute of Pharmacy. Hope yourself and your family are safe and healthy. Today, for the semester 3 and subject Pharmacognosy and Phytochemistry 1, we are going to learn about the new chapter, that is cultivation, collection, processing and storage of drug. This is unit 2 as per your syllabus. In this cultivation, collection, processing and storage of drugs, we are going to learn about the cultivation and collection, different stages of the cultivation and collection. What are the factors that affect the cultivation of medicinal plant? Like for example, altitude, temperature, humidity, plant hormones that play an important role, their application, polyploidy, mutation, changes, hybridization of the medicinal plants. So one by one we'll go to the topic. What is cultivation, collection and processing of herbal drug? Whenever a crude drug which reach to the market and pharmaceutical industry have passed through a number of stages, but the important point is it should be maintained as active constituent and responsible for the therapeutic activity. The stages are concerned for a drug because these drugs are useful for the mankind and the human being. So this chapter is concerned with the such parameter for the different processes. Cultivation produce improved quality of plants. It helps in selecting the species, variety of hybrids, and cultivation results in obtaining plants with maximum number of phytoconstituents. The main point is it's led to the industrialization in the country by the regular supply of plants, and it is also a useful tool for the research purposes. For example, if we go through the cultivation, ginger, turmeric and licorice can be cited as an example. If the cultivated plants are kept free from weeds, the contamination of crude drugs can be avoided. Collection of crude drugs from cultivated plants give a better yield and therapeutic quality. So the important point for the cultivation is the cultivation of crude drug, collection, processing and storage. Let's cover the cultivation. What are the advantages of the cultivation of drug? The first and foremost advantage is the improvement in the quality and the purity. We'll get a better yield and therapeutic quality. For each category of advantages, there is a particular example. For example, for better yield and therapeutic quality, whenever we collect the latex from the poppy capsule, we will get the oleoresins and different papaverine from the poppy. If this done by the experienced person and the pattern of incision is proper done, we will get a good quality of constituent and a better yield and the therapeutic quality. So this we can say that collection of latex is very important and minimizing the deterioration cardiac glycosides. It is also used in the cardiac glycosides. For example, cardiac glycoside digitalis can be achieved by a high skilled labor. So if the constituent can be cultivated in a proper way, we will get a better yield and a therapeutic quality. Regular supply of the crude drug can be obtained. Industrialization, modern technological aspects can be used. For example, mutation, polyploidy and the hybridization. Disadvantages, land, labor, money, high cost, everything is required. Now the important point is the organic farming. Organic farming is a method of agricultural production which avoid the use of synthetic products like fertilizer, pesticide, growth and regulator. And this can be widely used nowadays in a modern way of farming, that is organic farming. Microorganism with suitable pH, temperature and atmosphere, moisture, every condition is maintained in a well-behaved way. Now the methods of propagation. There are basically two different methods of propagation. One is vegetative, that is called asexual propagation. Vegetative propagation can be done by the vegetative part of the plant. For example, vegetative propagation for the bulb, example is allium for garlic, tuber for potatoes, rhizome for ginger. These are the different parts can be used for the cultivation. Now other vegetative method are cutting can be done, for example in sugarcane, layering in the jasmine plant, grafting, joining of new varieties produced by joining the two different parts. Advantages of the asexual production. The advantages are the asexual processes are these processes are genetically identified, and asexual propagation allow propagation of crops that do not possess the seeds. The crops that do not have the seeds can also be cultivated by this method. Now, another is the importance of this asexual. There are very many importance of the asexual propagation, like it is cheaper, easier, rapid method of multiplication. Plants like roses etc. do not have a viable seed. They can be propagated through this method. Next is the sexual. As the name suggests, plant raised from the seeds are the sexual method. Advantages are seedlings are long lived, seedlings are comparatively cheaper, easy to raise. This is the way how the sexual method is done from the seeds, the flower, anther. This is the way how the sexual process is done. Sexual method of propagation involves pollination, fertilization, seedlings, and multiplication of plants by using seeds can be done by seed propagation. It can be done by dormancy, rest period, seed viability. Different methods of seed improvement are the collection, exotic variety, hybridization. Mutation means change in genotype causes the mutation. Polyploidy means a set of chromosomes is multiplied and forms polyploidy with the different characters. These are some of the advantages and disadvantages of the sexual method. Next is the micropropagation. Micropropagation, as you have studied in the plant tissue culture. This method consists of the growing cell, tissue and organ in a culture. Small pieces of plant organ or tissue is grown in a container with a suitable nutrient medium under the sterile condition. Now, the important question that can be asked from this portion are the factors affecting cultivation. In factor affecting cultivation, I have given you 12 factors affecting the cultivation. These all are the names which you have to write, but the major factor you have to describe are the altitude, temperature, humidity, frost and snowfall. Means how altitude affects the cultivation, how temperature, how the rainfall, which type of soil is suitable for which cultivation, which type of light is needed, fertilizers, manures, plant growth hormones, pest, disease, preparation of land for the sowing, then sowing time should be proper method, type of planting, mixed cropping, alternate planting, strip planting so that the growth and constituent can be increased, organic farming, rotation of crops and variety of medicinal plants, interculturing operations, seed treatment, harvesting time. One by one, we will go through the factors affecting cultivation. First is the altitude. In the altitude, it is an important method. For example, certain plants grow at the proper altitude. Tea and cinchona require the altitude of 1000 to 2000 meter. We can see the example here. So these factors also affect the cultivation. Temperature is also an important factor. For example, cinchona requires 60 to 75 Fahrenheit of temperature, T to 70 to 90. These are the different temperature requirement. Humidity, camphor and coffee cannot withstand frost, whereas saffron needs cold climate. These are the different factor. Rainfall. For some plants, rainfall is very much important, but excessive rainfall can also cause a reduction in the secondary metabolite. Soil. The type of soil is also important. For example, the soil particle size plays an important role. Type of soil on which the percentage, clay soil, loamy soil, sandy soil, this also affects the color. Soil fertility, it is the capacity of soil to produce the nutrient required and balanced amount in the plant. So these are the different types of soil. Light is also an important factor, that in summer some crop grow well in summer and some in winter. Fertilizers, manures also play an important role. Chemical fertilizers, manures, biofertilizers, pest, diseases and the control. There are different types of pest. They are types of pest. The different types of pest infecting medicinal plant are fungi, virus, weed. Two things are there, pest and pesticides. Pesticides means which kill the pest. So types of pest, fungi, viruses and insects and weeds. This can affect our plants. Non-insect pest. Another is the methods of pest control. How to control the pest? By different methods, by culture method, physical method, biological method and chemical method. These are the different methods by which the pest control can be done. Agricultural method can also be used for the pest control. In this, we can use the methods like crop rotation. Crop rotation avoid the pest and disease of crop. Change in environment reduce the severeness of pest and disease. This is the biological control. Chemical control involve the fungicide, use of herbicides, fumicides, inorganic compound like zinc phosphate, variety of medicinal plant, proper seed treatment, preparation of land for sowing, proper land should be washed out, which is best land for the cultivation. Deep plugging is required. Sowing time, method and distance. Type of sowing, how to sow the plants is also different. Plant growth regulator, this is also an entirely important portion for the cultivation of the plants. Different plant growth regulators that can be used are auxin, cytokinin, gibberellin, ethylene and abyssic acids. These are the different plant growth. Different plant growth hormones have a different role. For example, auxin, the important example of the auxin is indole acetic acid, which plays an important role as a plant growth or hormone. Gibberellin, some produce shoot development, some root, some in equal amount produce the whole development of the plant. Gibberellin, cytokinin, example of gibberellin GA1, GA2. Cytokinin, Z18 is the example of cytokinin. Ethylene. So these are the different plant growth hormones and you should learn the structure. For example, for auxins, 2,4-dichlorophenoxyacetic acid is the example. IAA, IBA, indole acetic acid, indole butyric acid. These all are the plant growth or regulators named as auxins. Now this pictorial diagram will clear your concept that if auxin growth hormone is used, it can grow and destroy the receptor. Growth in the presence of auxin, the repressor is destroyed and growth genes is turned out. Repressor here, the repressor is not destroyed. Though the absence of auxin, the growth genes will not grow. So this is the example how the auxin or the plant growth hormone play their role. Function of the auxin in the cell elongation, also in apical bud suppressor, growth of lateral buds. Cytokinin causes cell division. The example is kinetin, stimulate cell division. Kinetin, Z18 are the example of cytokinin. Auxin alone will give no divisions. With cytokinin alone will give cell have no change. But auxin and cytokinin will give the normal growth. If auxin is greater than cytokinin, then shoot development takes place, and if auxin is in more amount, then root development takes place. If auxin is in less amount, then shoot development. Abyssic acid, this is the abyssic acid and the example is abyssin 2. Please note the structure is also very important to draw in exam. Gibberellic acid. Gibberellic acid helps in elongation of stimulating cell division, internode elongation. Gibberellic acid is the example of gibberellic. Ethylene, CH2CH2. Now, hormones are the biochemical that regulate the plant growth. Auxin and cytokinin also play a very important role. Now comes the collection. For the collection, what are the important points to be required? Skilled labor, season, age of the plant, time, stage of the plant. Collection of drug. Collection of drug is also important parameter as collection should be done at the proper time, at the proper way. Harvesting, general rules for the collection of the drug. For example, root and rhizome have different time of collection. Bark, leaves, how to collect the flowers, method of collection by the hand, by machines. And as per the WHO guidelines, medicinal and herbal drugs can be harvested when they are the best possible quality, when they have the best time for the important quality phytoconstituent. And these are the important points to remember. Drying is also important parameter. Some plants are dried at open air in the sunlight, some drying in oven, some in vacuum dryer. Nowadays vacuum dryer, spray dryers are used for drying the latex, for drying some enzymes obtained from the plants. So drying is also a skill. You should know about how to dry the constituents. Storage is another important factor. How to, as you all know, crude drug contain the volatile oil and the phytoconstituent. They can be stored in a container. They can be saved from the microbial growth. They can be easy to have their phytoconstituent, to save their phytoconstituent. So there are the different ways for the storage of the drug, packing of the drug and preservation of the plant material. Preservation of the plant material means, so the active content, the main purpose of crude drug is to maintain the active constituent or active compound during the transport or the storage till it get formulated. Nowadays, as you all know, different herbal formulations are running in the market with the proper concentration of the phytoconstituents. These are the important questions that can be asked from the cultivation and collection chapter. References: So cultivation, collection, processing and storage of crude drug. The main important points to cover are the factor affecting the cultivation, different types of plant growth regulators used in the cultivation and collection. Thank you.